Hi, thanks Marcus for taking the first part and for helping me put all this together and convince Jonathan that it was a great challenge. I mean, I have to admit that when I started this, I had no idea we were going to be able to bring it to a certain point, and there has been, the path is not always direct, but the path always takes you to Rome. So this is kind of how we did the scoring. So we wanted to score first the distance to the parameters, and so because of course parameters have different values and they can vary a lot, we just did the log of the distance, the squared log of the ratio of the distances between the true value of the parameters and the estimated value. So this for the 30 parameters that people had to predict. And there was also here's the prediction accuracy. So this is based on the phenotypes and distance to the phenotype. So basically, just a distance from the actual values of, let's say, mRNA levels or protein levels or metabolite concentration, and then divided by the variance. And this was the value of the final score. So we calculated p-values from these distances, and then we had a final score. So actually, these distances were given to participants every time they would submit a model to the cloud, so they could have an idea of how well they were doing. And you'll see that the fact that we gave the parameter distance, of course, brought some situations that I'll tell you in a second. So just briefly, if my slide comes up. There you go. Okay, so we had 100 participants that at least subscribed to the challenge. Wide diversity of countries. I guess Colombia being predominant for some reason, and I'll tell you a couple of anecdotes about them. What we really liked, and again, this was really one of the purposes of the challenge, is that yes, there were a lot of computational biologists, but even some biologists tried to just try to use the model, and that was really the idea of the challenge. We always want to try to bring the difficulty of the data sets in DREAM or the challenge or the model you're using, so that anybody can try at least to put a hand on it. And I think that's a really important thing, and so it's very encouraging to see that some biologists try that. And actually, one of the winners is someone who has never really, one of the best performers had never really worked with these kind of models or metabolism or anything related. And of course, a lot of the people that have free time or a lot of time to give for interesting projects are grad students. So. Okay, so this is a little bit just the statistics of how the competition went. So as you can see here, these are the number of submissions that participants did, and these are the number of participants. And as you can see, so there were a few teams that were very active. So around 8 or 9 teams, and then a few teams just tried a few times. So out of the 100 that really subscribed to the competition, around 10 were really active. So it's not as many people as in the other challenges, but we were happy that we brought people and people were really using. And as you can see over time, people were submitting more and more towards the end. So there was a lot of activity, and Gustavo already gave the number, but at a certain point there were up to 300 submissions a week, so it was pretty active. So this is how the participants did. This is the parameter distance, and this is the prediction distance. So as you can see, something that we had seen already in other DREAM challenges is that the parameter distance is not always correlated to the prediction distance. So again, the prediction distance is the phenotype, and the parameter is what you would think is what's bringing the phenotype. So you would somehow think that these are more correlated than what you see here. You see here that there's a huge variation in the distance of the phenotype compared to almost a single parameter. And you can see here that somebody did really, really well, so 10 to the minus 20 in terms of parameter distance, but still that the phenotype distance was not so good. And you can see again that people, as time went by, people okay, this is not working again. So it's the same. Well, you can basically see on the green line that as time went by, participants were doing better, and really at the end there's like this massive fall in terms of the parameter distance, but it's not the same case for the performance, which was basically more like a linear downhill. So again, this is the set of the distribution of the histogram of the parameter distance. So you see a lot of where just basically around the initial distance we gave them, which was around 10 to the minus 2, and here is the team that did very well in the parameter distance. And here is the phenotype, and you see the phenotype is actually quite diverse, and it's more not as spread as the parameter distance. This is more related to actually how participants did for each of the parameters. So here you have the parameters separated by the 3 groups, as Marcus said, so RNA pole, RNA half-life, and the reaction kcat. And in green, you see the ones that were modified, and in blue, the ones that were not. And you see that, of course, the ones that were modified, there's more variation in terms of the predictions up here. So this is just the log ratio, the distance I showed you. But you see that the reaction kcats were actually harder to predict, where you see more variability here. And you see on the plot down here the correlation between the distance and each of the parameters, and you see that, again, the reactions were the ones that were the most correlated, and so hence more difficult to predict. Again, this is just the correlation between the prediction distance and the parameters, and you see that the kcats were the hardest to predict and were the ones that were the higher up here. So really the reaction rates were the hardest to predict. Okay, so as we said, there's people could purchase data based on the modified model, and what you see here is that actually people didn't use a lot the data, but you have a team, which is a Team Colombia, which had 10 members, all of them registered and downloaded all the data. What's strange is then they didn't submit anything, but they were really eager to get the data. So I wonder what happened with them. If anybody knows them. And then there was the team New Dream, which was very active. And another team that downloaded some of the data. And basically, initially they concentrate in getting some of the single cell dynamics, because that's in a sense how we had designed the challenge. The idea is that, in having such a complex model, you wanted to have things that were a bit simpler to infer to, and so we thought that the cell division time was an easy parameter to try to fit initially, and so people seem to have bought some of that. And we also indicated that we were only going to modify parameters on the metabolic submodel of the big model, and so people were trying to use some of this data. But again, as I said, data didn't really seem to be so essential for this challenge. So these are some of the best performing teams. So Team Crocs used the maximum likelihood. Wholesale modelers were using random forest, and we'll hear about these 2 methods. And the teams ICM Poland, and New Dream, actually what happened was that because we were giving the parameter distance, they were just using that, right? So of course that, you just use it 30 times and then you get the result of how you modify the parameters. Although it's not so simple because this is a stochastic model and it has some, similarities, but the question is, and I'll show you this briefly, in a second I'll comment a little bit more on that, but here are the 2 best performers that you're going to hear. So these are the number of submissions. So Crux had 99 submissions. They actually, the result they got was by using gradient descent, but a lot of the techniques they used before that didn't use gradient descent, and I hope that's what they're going to be talking about today, because we don't want to hear about gradient descent. And wholesale models, as you can see here, they didn't do so well in terms of the parameter distance. They're pretty high. They actually, one of the comments was that their submission actually did worse than what we gave initially. But what's interesting is that they did pretty well on lowering the distance from the phenotype. So they came down here, and here's where Crux is, and this is the initial distance. So they were really able to infer just from the data to get a better distance to the phenotype. So actually, that's a great result, and we're really happy about that. These are just some of the paths that people took. So this is the prediction distance, and this is the parameter distance. And And basically, for example, in this team tried a few things and then realized that by gradient descent that we could get a much better result. And because they saw that the other teams were also using that, then just the competition kind of just drives you and you want to win it, and whatever method you use. And so basically what we did is that we decided to separate people that of course use gradient descent from the ones that didn't, because it's not so fair. But so here you have over time just over the number of submissions, how well people did in terms of parameter distance, and you see that in red you have the team wholesale modelers. Again, they didn't do so well with the parameter distance. In dark red you have Team Crocs, and I'll show you here. So Team Crocs here are the ones that did better, and over time they really went kind of down with the parameters. And finally, here are the prediction distances, and as you see, there's this Team New Dream that's, it's the ICM Poland that really, I think they were trying to do better, but they just went from, worse to good. I don't know exactly what they were doing, but they seem to be really trying a lot of different things. But as you see here, the Team Crocs initially, stayed kind of constant, and then at the end had a, pretty good prediction. And here again, Team Wholesale Modelers. I don't know why they stopped submitting, but I guess probably because they were not converging, having a better prediction than what they had initially. Okay, so these are just the two basically final submissions from two different groups: people that were using the parameter distance, people that were not. And you can clearly see that also in terms of the number of submissions they had. I mean, this team had 290 submissions. Crocs, although they used the parameter distance, which was, a few, half of the submissions they could reach for a much better distance, 10 to the minus 19, so 8 orders of magnitude better than these guys. And Team Wholesale Modelers, again, from people that were not using the parameter distance. So we have a few prizes over the competition. So one of the aims is try to publish these methods in a paper for PLOS Computational Biology, but also PLOS gave us some T-shirts and some mugs and things like that, and MATLAB gave some licenses. And it's kind of funny because somebody criticized the fact that the model is run on MATLAB, and so that you need, you can only basically solve it with MATLAB. And we were giving away MATLAB licenses, so I guess it's kind of like, if you win, you get a MATLAB license, which you need to anyway solve the problem. So. But anyway, it's always useful. Okay, so the code of the model is available. We actually enhanced a few of the things to try to make it as easy as possible, so we hope that people will try to keep using it. And these are just the people that participated. I think it was a great fun to see people excited about a complicated problem and trying to make it as easy as possible, because I really think that these kinds of models, and me being an experimental as well, being a little bit in the two fields, I was really impressed by the results of these models. So I think really the combination of the model with experiments is going to be the future. So thanks a lot.